There's a revolution happening in medicine right now, and it's absolutely incredible. We're talking about drugs that can target diseases we once thought were completely untreatable. But as we're about to see, with this amazing new power comes a whole new level of complexity and a whole new kind of risk.
Okay, so to really get what's happening, you have to appreciate the just staggering leap in scale. On the left, you've got a classic drug like aspirin. It's a simple, predictable little molecule. But on the right, that's a modern biologic drug. The difference in complexity here isn't like comparing a brick to a bigger brick. No, it's like comparing a single Lego brick to a full-scale Lego Death Star. We are playing in a totally different league of biological engineering.
And this isn't just about being bigger. It's about what these drugs fundamentally are. We are officially in the era of the living molecule. So what even is a biologic drug? Well, unlike a typical drug that's synthesized with chemicals, a biologic is made by living things. We're talking hamster ovary cells, bacteria, you name it. The two things you absolutely have to remember are its super complex 3D shape and this thing called intrinsic variability. Basically, because they come from living systems, no two batches are ever 100% identical. Just like no two tomatoes from the same plant are ever exactly the same. I think this quote from Dr. Yannick Bardie just nails it. Here, we don't make a molecule, we orient a living process. Think about that. We're not following a fixed chemical recipe. We're guiding a biological system. We're acting more like gardeners, kind of coaxing a living thing to produce what we need. And, just like in a garden, you always get tiny variations. Now, all this amazing complexity, it comes with an equally jaw-dropping price tag. Some of these therapies can run over $1 million for a single treatment. And that, as you can imagine, puts this enormous pressure on our healthcare systems to find a cheaper way to do this. And that cheaper way has a name, the biosimilar. You can think of it as the biologics' non-identical twin, and it was created to bring some much-needed competition and lower those eye-watering costs. So a biosimilar isn't your average generic copy. The official term is highly similar. It has to prove that it's so close to the original drug that there are no real meaningful differences in how it works or how safe it is. But here's the crucial part. Highly similar is not identical. Not even close. And this brings us right back to that idea of a non-identical twin. A generic drug, like for aspirin, is an exact chemical copy. Easy. But you can't exactly photocopy a living process. So a biosimilar is made through its own unique manufacturing process to be a really, really close match, a controlled but still non-identical twin. And this is a really key point. The whole goal of the testing for a biosimilar isn't to prove all over again that the drug works. We already know it does from the original. The goal is just to prove similarity. This makes everything way faster and way cheaper. But it also means we're putting our trust in a different kind of scientific proof. And this whole new world of similar but not the same leads us to a really critical question of safety. What happens when one of those tiny, acceptable differences ends up having huge, completely unexpected consequences? This isn't just a what-if scenario. It actually happened. Back in the early 2000s, a company made what they thought was a super minor change to the formula of an EPO product. That's a biologic that helps your body make red blood cells. Just a tiny tweak. But then, reports started to trickle in. Patients' own immune systems were freaking out, creating antibodies that didn't just attack the drug. They started attacking the body's own natural EPO. The result was a sudden spike in cases of severe, life-threatening anemia. The lesson we learned the hard way from the EPO crisis is that with a biologic, the risk isn't just in the drug itself, it's in the entire fragile journey it takes. The manufacturing, the formula, even the type of rubber stopper on the vial can matter. One tiny, seemingly harmless change can set off the immune system. That's called immunogenicity. And it proved, without a doubt, that we have to keep watching for problems long after a drug is already on the market. So this brings us to the big, flashing question for healthcare today. If one little change in one product could cause such a disaster, what are the potential risks when we start switching a single patient between the original drug and maybe 2, 3, or 4 different non-identical biosimilars over the course of their treatment? This isn't just about one molecule anymore. We need to zoom out and look at the risks that are building up across the entire system.
A really helpful way to think about this is something called the stamp model. It says that huge accidents don't usually happen because one single part fails. They happen when a really complex system slowly loses control, because all the safety rules that are supposed to hold it together get weaker and weaker over time.
To make this real, just think about disasters like the Challenger explosion or Chernobyl. In both of those cases, it wasn't one sudden surprise failure. There were known problems, there were engineers raising alarms, but there was also this immense external pressure to launch, to meet quotas, that led people to bend the safety rules. And the result was catastrophe.
In the world of biologics and biosimilars, we see those same kinds of pressures. You have massive economic pressure to make things cheaper. You have temporal pressure to approve drugs faster. And you have political pressure to get these drugs to more people right now. And each of these forces, even with the best intentions, can start to quietly erode the safety measures put in place to protect patients.
So how on earth do we manage all this? How do we get the benefits of cheaper drugs without letting the system spin out of control? Well, the answer seems to lie in building a totally modern way to govern it all, powered by data and artificial intelligence.
The way forward really has 4 main steps. First, total traceability. We have to know, down to the batch number, exactly which drug a patient got and when. No exceptions. Second, we have to collect and analyze the data from patients being switched between multiple products, because that's the new frontier of risk. Third, we need to use AI to constantly scan all that data, looking for those super faint early warning signs, the weak signals a human would almost certainly miss. And finally, all of that gives us smart governance, where decisions are made based on real, up-to-the-minute evidence.
Like Dr. Bardie says, without traceability of the lot and brand, pharmacovigilance on biosimilars is just astrology. I mean, think about it. If you can't trace a problem back to its source, trying to figure out what caused it is literally just guesswork. You're flying blind. Good data is the absolute foundation of safety here.
Which leaves us with one last thing to think about. The era of the living molecule is giving us these incredible medicines, but they exist inside a system that is under enormous pressure. And as we push forward, we have to keep asking ourselves, when a system this complex starts to wobble, do we even know who's really in control?